Oh, hi there, William. It was good to meet you today. So, as promised, we're just going to have a wee look at the microbiology in your soil. So, this was the one southern field that I was sampling. I can't give you the parcel number, but I will show it. I'll highlight it on a map for you and send it to you on WhatsApp. I think it was just oats, or it might have been wheat. It had a lot of residue. It had just been cut recently, so there's stubble and residue. Um, so this is like a sample from across the field. And this is diluted at about a 1 to 10 dilution. So 10 parts, 9 parts water really to 1 part soil sample. Like we'll just have a brief little scan through this soil, and I'll tell you I'll point out anything interesting, try and give you some soil facts as we go. Um, so yeah, when we're performing like some sort of qualitative soil assessment, we're kind of looking for functional groups basically, and the presence of various trophic layers of biology, both single-celled and multi-celled, prokaryotes and eukaryotes. And this is just to ensure that the nutrient cycling circle is functional, basically. So without, for example, all the nematodes in their various forms, and all the protists and bacteria and fungi, we're going to have an incomplete cycle. The nutrient loop is going to be broken somehow. So we know that plants can only get their nutrients in a plant available form, either through inorganic fertilizers, synthetic fertilizers. But the way that they do it in the wild is getting it through the microbiology that they attract when they put down their carbon chains from photosynthesis around their root zone. I'm sure you're familiar with all this stuff, but very quickly, essentially, the plant harvests liquid sunlight in the form of carbon, and it releases it in various forms into the rhizosphere, and that is to attract very particular species, and actually it goes all the way down to the individual level, the individual bacterium, fungi, protist, nematode, that plant has a symbiosis with. So it can actually it can create a chain which will act as a stimulant for an individual bacterium, we now know. So it's this very, very complicated communication, quorum sensing, the fancy name for it, that's happening in the soil 24 hours in the day. And also there's been some quite fascinating, I'm trying not to get too sidetracked here talking about soil, but there's been some very interesting work looking at the rhizophagy cycle. James White, you should have a look at his lectures. And it turns out that, well, as far as we know, all plants are effectively predators, which is quite fascinating. So not just the insectivorous plants that grow in swamps, but actually wheat plants and all terrestrial plants are actually predators, and that happens through something called the rhizophagy cycle, where they effectively farm microbes internally within their body, and they can fix nitrogen this way, and they can farms and clone some of the microbes that they require for growth, but they can also cull some and absorb their nutrients directly in their body. But anyway, I'll let James White describe. If you're interested in that, you should look him up. It's really quite fascinating. So this is a really, really, really quick little overview scan. We're at 40 times total mag here. So here we'll be able to see arthropods, mites. We'll definitely be able to see nematodes here at this kind of magnification. Sorry about this mottling effect. Let me just get that fixed for you. It's very sloppy microscopy from me. There we go. We'll be able to see nematodes at this point. When it comes to nematodes, I don't know what your relationship with nematodes is like. If you ever have any sort of root canker nematodes and clubroot nematodes or anything of this nature. But often the best way to combat these these pests, don't like using that word, but we'll call them pests for the sake of simplicity, is actually nematodes, but of a different species. Lots of predatory nematodes. Predatory because they actually feed on other nematodes are the way to go, a very effective biocontrol. I'm not talking about buying them off a company, but when your soil quality improves dramatically, these nematodes will naturally colonize. And they actually preferentially pick out disease-causing nematodes or pest nematodes and predate them, which is quite fascinating. So that's really rather handy. Can definitely utilize that. So I'm not really seeing anything. They're very obvious. I'm sure you've seen them before. They tend to thrash around a lot. But I'm not doing a proper survey here. I'm just skimming through quickly just to give you some interest here. So that's gone through pretty much the whole slide here. So we'll just zoom in and see if we can see anything, especially like fungal structures, particularly interesting. And I'll just zoom right in here. So this is 400 total mag, so 400 times what human vision is. Now we can start to see bacteria, we can start to see things moving. And this is an 18 by 18 millimeter cover slip, by the way, so it's really quite tiny. So all those things that are gyrating around, they're all bacteria. The things that are almost perfectly circular are cocci, and they approximate something like 1 micrometer in diameter. Micrometer being 1,000th of a millimeter, so very small indeed. So that's the kind of scale you're looking at here. The transparent window pane-like objects, those are silica, little particles of sand. Some of these more coloured objects, like let's find a big one here. These are your aggregates here. So the things that are kind of honey coloured and they almost look a bit like saliva, like that one right in the centre. That's probably going to be largely composed of ulmic acids, fulvic acids for the slightly lighter honey coloured aggregates there. The really, really dark spots will be humic acids, but it doesn't look like there's a huge amount of humic acid present here. It looks kind of, this mineral soil looks quite mineralized, so it doesn't look homogeneous, which is kind of what you're looking for when you look under a microscope. You want everything to be pulled together. Soil's kind of fractal, so what you see, you want to see a kind of crumb structure almost underneath the microscope that you would do when you're doing some sort of VES kind of assessment. So there's a big aggregate there. But you don't want to see too many individual minerals, really. These minerals should be getting incorporated into these aggregates to be broken down by primarily fungi. They've got the enzymes capable of breaking apart these kind of matrices and accessing the nutrients held within them. There's a spore there, or possibly a diatom. You see in the center the thing with like two, there you go, the thing with two blobs right in the center there, that is some sort of fungal spore. I couldn't tell you what species, only that it's not Basidiomycetes, but I couldn't tell you any more than that. So yeah, what we're looking for is fungi. We're wanting to see maybe one per, maybe 2 fields of view. There's this correlation that I really kind of briefly mentioned where if you can get your fungal to bacterial ratio sympathetic with where your crop species is in succession, or its wild relative would be in the wild, that's when we get the real gains in terms of yield, in terms of nutritional complexity, in terms of disease resistance, drought resistance. So for most vegetables and most grain crops, we're looking at something like 0.5 to 1 or 1 to 1, an even ratio of bacteria to fungi in terms of their biomass. And I'm not talking about species, but we also want to see very, very diverse species as well. There's some actinobacteria there, these little strands. Actinobacteria can be facultative, so they can be indicators of changing oxygen conditions. And we want our soils to be predominantly aerobic. We don't want them to be entirely aerobic, because that's obviously when things start getting oxidized. But by aerobic, I mean something like 4 to 6 parts per million oxygen. So, it's lower oxygen levels than you would find just out in the atmosphere. That's a big bit of ulmic acid there, I believe. So that's quite nice. And typically, especially if you're you're doing all your carbon audits at the moment, I know you want to see really high fungal numbers, because those are the things that are going to sequester vast quantities of carbon for you in the soil. When they respire, on average, this is talking about so-called beneficial fungi, so not oomycetes or pathogenic fungi, although that's a whole different conversation. But anyway, we'll not try and get too complicated here. Beneficial higher order fungi, they respire only about 20% of the carbon that they ingest as CO2, and with bacteria it's about 80% they respire. So they give off a lot of CO2, and classically, like at least what I've seen in a lot of Scottish soils, agricultural soils, is that they're very, very bacterially dominated. And this is just an artifact of farming, really, or historical farming. I know you're not doing inversion ploughing anymore, but fungi are very, very sensitive. And, any soil disturbance, they're going to be the first ones to go. Bacteria are going to be the first ones to colonize and effectively feed off that. If you are doing inversion ploughing or ploughing, there's going to be a bacterial bloom because of all the necromass, the dead bodies of the fungi and bacteria that were immediately destroyed. And that's why you get so much gassing off of CO2. So yeah, we want to see high fungal populations, and like I say, that's sort of like very roughly translates as about seeing 2 hyphae per field of view. So we would have seen a lot of hyphae by now if we had a good fungal presence in the soil. So that might be something to work towards, or see how you can change management to affect that. I think really I don't know what your sort of chemical use is like, or chemical applications on the farm, but I think that's where you're going to see the biggest gains is when if you can highly reduce or even entirely stop chemical additions to the soil. That's when the microbiology is going to really start thriving. And also, when you have these bacterial ratios to fungi about 1-to-1, that's when germination of higher order plants that are higher in succession is selected for as well. So the weed bank won't be stimulated to germinate anymore. They say what is it? 1 year's ploughing, 7 years weeding, or something like that. And that's also because weeds, again, because of quorum sensing and because of the communication between microbes and plants, they're stimulated to grow in conditions where there's very bacterially dominated soils, to try and heal them, if you want to make it sort of anthropomorphize it. But that's their niche in ecology, the weed, as it were. Not really weeds, just early successional plants. Anything that races to any annuals that race to reproduce, go to seed as fast as possible, and have a large presence in the seed bank in your soils. So yeah, I would say the lots and lots of bacteria. Let's just have a pause here, and we'll actually see, because we're of course looking for diversity in bacteria as well, and we're just doing that not by speciation, like we're not going to do DNA analysis or anything like that. And I think DNA analysis, it definitely it definitely has its place, but what we're really looking for is not species present. We're looking for active organisms, right? Because that's going to be the most informative to a farmer as to his management. So DNA analysis or phospholipid fatty acid analysis is only going to give you a huge, massive reel of a list of all the DNA that's sort of in the soil, but those organisms might be in dormancy or cryptobiosis, so that's not going to be so helpful to inform you. So I think direct macroscopy, I think if you feel suitably inspired, definitely investing in a relatively cheap £200 to £300 microscope is really quite fascinating. So yeah, just having a wee zoom in and out of this sample here and looking for different shapes. So bacillus shapes, then the more classic looking bacterium. Cocobacillus is kind of a mix between a bacillus and a cocci, which is the round one. And you get sort of lactobacillus and actinobacteria, which are more filamentous looking, especially the actinobacteria. So yeah, we're seeing some diversity in the bacteria. Certainly seeing cocci and a few bacillus, but apart from that, not too many shapes. No pathogenic fungi, bacteria though, so that's nice. Wouldn't really expect to see them though. That'd be very, very strange if we did. So yeah, and then of course the protists as well. They play like a really vital role in making nutrients from the bedrock material, basically, of your field available to the plant, just through predation. So your average protist will be eating about 10,000 bacteria a day, so that's really quite a large number. And they will be expelling the excess nutrients that those bacteria had, that the protist does not require for itself, out around the plant's roots and making that available to them, be that nitrate, phosphate, magnesium, calcium, anything, any of your macro and micro trace elements. That's just good. That's why I was asking about the total nutrient pool rather than just total available or soluble nutrients. The total pool of nutrients that are immobilized in your soil, that can be really informative, because not all soils have all nutrients available in their bedrock profile. But essentially, but most soils do in the world. Most soils do have all the nutrients that you could possibly require for a plant to grow well. And it's just for these microbial interactions with the plant roots, when that's functioning at high rate, then you won't need any additives, any external inputs that are synthetic anyway, or made by industry in some way. So yeah, very interesting. Can see, yeah, it's definitely actinobacteria are quite prevalent, but I haven't seen any fungi so far. This is of course not necessarily representative of your field, but let me just have one final scan and see if I can just spot one zoomed all the way out, and then I can show you what a fungal hyphae would look like. That could be something of interest there. No, I think that's just a bit of organic matter, or plant root, or plant hair, I think. A root hair. Yeah, root hair, I believe. So yeah, I think, I don't know, I mean, it it's like I was saying, it's interesting to experiment, and I think you need to obviously be convinced of all these things in your own mind, but certainly be interesting to try the diverse cover crop mix as diverse as possible. You can't really go wrong. It's just the question of terminating it. How do you terminate it if you're not going to use if you're not going to burn it off with chemicals? That's obviously the $1 million question. But presumably a lot of plants, at least hopefully they can make it at least through most of the winter and then get killed off in the frost. But yes, 17 species cover crop. I mean, if you're going for an experiment, why not just go for a 30 species cover crop, And if it's only a very small area, you're not going to spend a huge amount of money on it. I think these kind of tests are completely, they're really invaluable knowledge for other farmers. So, any farms that are willing to do a bit of experimentation, it's great knowledge for other locals. And compost would be the other one. But that's a science and an art, and it needs to be monitored closely if you're going to do it serious. This is for serious agriculture, and salaries depend on this, so it needs to be done correctly and well planned and well thought out, and again have a large pool of ingredients that are getting added to it, and yes, indeed, and making sure you got all the ratios correct and everything, and it's aerated, etc. And applying it correctly, through drip irrigation probably when you're seeding. I'll send you a link to John Kempf, because he's a wealth of knowledge if you're interested in any of this stuff. At some point it'd be nice to come out to your farm and actually do some comparisons, and I'll give you a full bioassay if you want, of like a few of your fields, and especially if we can compare one field to another, something that can consolidate in your mind, sort of maybe like one field that's performed particularly well, and maybe you're not sure why, and one field that hasn't performed as well as you would like. See if it's the microbiology might give you some answers there. But yeah, good to meet you, and take care. I'll see you soon. Come down to the farm, by the way. All right, bye.